Title of my talk today is Integration Across Disciplines in Clinical Microbiology and Urology. So here at the Vanderbilt P20 Center, I lead a pathogenesis group in microbiology, as well as serve as a clinical microbiologist. And the theme of this, the next few minutes, is to talk about the need for greater integration across clinical urology practice, clinical urology research, and clinical microbiology. But first, I think it's really important to define exactly what we mean when we're talking about clinical microbiology. So the burgeoning need for microbiology research in translational urology is unquestioned. So, of course, urinary tract infections remain one of the most common clinical conditions in day-to-day practice, and their pathogenesis remains a topic of very cross-disciplinary study. But even beyond that, over the last decade, there's been an increased appreciation that urine is not inherently sterile, and that the urobiome of patients, even those without frank urinary tract infections, is a real thing and something that can contribute to diverse urological conditions, from cystitis, even when we're talking about non-benign conditions like cancer. So really, the idea that this is the idea that urologic research and microbiologic research need more integration, not controversial, but specifically, I'm talking about clinical microbiology laboratories, the laboratories that support patient care through the diagnosis of patient specimens. So what is a clinical microbiology laboratory, and what's the difference with a research microbiology laboratory? I like to joke that if you see a clinical laboratory versus a research laboratory, in some cases it's obvious, but stereotypes of lab tidiness aside, they have different fundamental missions. Research laboratories, of course, are driven to expand the state of knowledge and to find novel associations and, importantly, causal mechanisms. By contrast, clinical laboratories they're tasked to leverage existing knowledge and technology to provide predictive and actionable value on an individual patient level. On a practical level, the finances, as we all know, of research laboratories are often dominated by the bottom line of grant cycles and the ability to obtain publicly funded support for our work. By contrast, in the clinical laboratory environment, the finances are very much dominated by reimbursement parameters. And the research, the R&D in the clinical world is much more guided by industry relationships, either industry-sponsored work or work produced at the industry themselves. And then, of course, at a level of quality, of course, scientific research is accountable to high standards of rigor, reproducibility, peer review. But in the clinical sense, while we're held to those standards, there's also very strict legal and regulatory parameters. And whereas research laboratories have to have adaptability of their methodological approaches to adapt with ongoing results, clinical laboratories have very fixed constraints to deal with because of those regulatory situations. So what are we talking about then in terms of the relationship between urology and clinical microbiology? I liken it a bit to a person on a bucking bronco, with urologic research and uromicrobiologic research being the horse, taking clinical microbiology a bit along for a ride. We're making a lot of ground, but it's a bit tumultuous, and there is a potential to get bucked off at times, and there is a potential for a bit of a Wild West dynamic as well. So what are some examples here where emerging science and technology and microbiologic research in the urologic sense is taking clinical diagnostics for a ride? Well, let's start talking about traditional UTIs. This, of course, are the guidelines that existed for decades about how UTIs are diagnosed through semi-quantitative culture, where we look at the ability of organisms to grow under very common aerobic conditions, and we use both the diversity and the prevalence of organisms to determine if a urinary tract infection exists. Unfortunately, as we know, some of these guidelines, while still in clinical practice, have their flaws. A lot of research has come out saying that even frank urinary tract infections can exist with CFU per mil levels far below what is traditionally thought to be clinically reportable. Conversely, there's also this issue of emerging uropathogens and the idea that organisms that aren't traditionally thought to contribute to infections can in fact contribute. Many of these organisms likewise are more fastidious and blur the line between dysbiosis and frank infection, returning to the microbiome theme. One thing that folks has asked is, well, if culture is good, can we go further, and can't PCR be better? And I say this is not a hypothetical. There are clinical laboratories, commercial clinical laboratories, who have brought this into actual practice, marketed it to urologists, and it is used. So instead of going from a situation where you're doing culture, can you do parallel qPCR against a whole bunch of targets and utilize that to overcome some of the challenges? You could look at fastidious and non-fastidious organisms together. You don't have to worry about traditional culture conditions. There's some advantages to that, but there's also some challenges. What about things like specimen quality and the need to look at things like urethral contamination? What about the definition of relevant uropathogens to target? With these emerging concepts of dysbiosis, what you actually target isn't always straightforward. And of course, this leads to a situation of billing constraints and even overbilling, where you're doing lots of molecular CPT codes for what potentially could be done with a single culture. And then, of course, maybe most importantly is the need for clinical context. I bring up here recent guidelines of asymptomatic bacteriuria to put out, even without the concept of a urinary microbiome, the interpretation of a clinical urinary result all depends upon the context of the patient, both their clinical history as well as their particular symptomatology. So can we go a step further? Could we utilize clinical use metagenomics and urobiome profiling as a diagnostic tool? I mentioned that this could potentially be used in two ways: for catch-all identification of potential uropathogens, where we utilize next-generation sequencing and broad-range amplification as an all-in-one to have to avoid the need for dozens of individual PCRs. Or we could talk about true diagnostic profiling of microbial population structures, true microbiome-based diagnostics. Some similar challenges still apply: appropriate ordering, context, defining actionability, and even billing parameters. For these, there are no CPT codes out there that exist for reimbursing these. But there's some really cool opportunities too. Recently, some consensus statements came out about urobiome research and about defining things like specimen collection, site of collection, bioinformatic parameters, and the need to systematize this across urobiome research. Going forward, we now have to expand this consensus from a research to a clinical diagnostic setting, even defining basic parameters in diagnostic practice for which microbiome-based testing is sort of a square peg in a round hole. Parameters like analytic sensitivity, diagnostic sensitivity, even normal reference ranges themselves don't apply to microbiome-based analyses as they necessarily do to a detected, not detected analysis. So lots of work to do there. At the same time, I think there's a lot of potential for exciting growth. And I think reversing the roles, the clinical microbiology laboratory can also be the horse, so to speak, driving urologic science forward through a couple of really unique resources that we have in the clinical environment. Most importantly, we are a source of vast wild-type bioresources. We have a massive diversity and number of urologic specimens that come through thousands and thousands a year, one of the most high prevalence benches in the laboratory. More importantly than that, even we have linkage to electronic health information on a paired level. We can link individual patient data with urologic specimens in order to draw conclusions. And overall, this allows us to move beyond simplified experimental models of pathogenesis, utilizing one-off model organisms and models of in vitro or animal infection, to a more holistic concept of what's going on the species level. This is what we do at Vanderbilt here with our Vanderbilt Urinary Tract Infection Repository, our P20 Center, where we've created a logistical pipeline for banking all of these urological isolates that arise as the course of clinical care in concert with our Institute for Clinical and Translational Research. Here we take our regular clinical workflow, which is we get a specimen, we analyze it, this information goes into our LIS and EHR, and then finally goes into a dedicated compendium of research electronic data at the institution. We actually further interface this data with upstream instrument analysis, but more importantly, we banked these organisms and created a framework for de-identifying them to us, but still leaving these organisms linked in a queryable manner to all of the electronic health data on these patients. We've also done this for microbiome specimens, looking forward to potential further avenues as well. Ultimately, what this allows us to do is you could query an organism based on taxonomy, resistance, ICD-10 codes, whatever you want, patient parameters, You could get the clinical isolate. You could do more experimentation on it. You can get the patient phenotype from our research data warehouse, and even the patient genotype, because a lot of these folks have banked human DNA specimens from our BioView repository, which unfortunately I don't have time to go into. The net result is the possibility for association level genome-wide, phenome-wide, or other multi-omic association studies on a very vast level. So where are we with this is we have all diagnostic urinary isolates banked and linked to our clinical records since we've instituted this P20, and we've been able to apply these diverse hypothesis and discovery-driven projects, which I'm actually not going to go into. I will leave that to be continued tomorrow by one of our fantastic students, Grace Morales, who will be talking about this for more. So on that, I'm going to sign off. I apologize again for the initial technical difficulties. I want to acknowledge all of the folks who contribute to this, including especially the co-PIs on the P20, Maria and Doug, as well as various sources of funding. And with that, I will sign off.